All right. Hello, everybody, and welcome to our annual Nasrallah lecture for 2025. We have a distinguished speaker today, Dr. Anissa Abi-Dargham from Stony Brook University. And before I introduce her, I just want to remind people, if you're watching this on Zoom, your cameras and microphones are off. We do encourage people to ask questions or comments, post comments, but do that in the Q&A function rather than in the chat, which we reserve for the panelists and the interpreters. And also I want to remind people to fill out the evaluation at the end, or the QR code will be posted up here and sent out in the chat as a link as well if you're on Zoom. Okay, so our speaker today, I'm really glad that we were able to get her to come here, Dr. Anissa Abi-Dargham, who is a SUNY Distinguished Professor of Psychiatry and Radiology, as well as the Laurie Endowed Chair of the Department of Psychiatry. She has an endowed chair, the Laurie Chair, and is the chair of the Department of Psychiatry and Behavioral Health, and the Associate Dean and Associate VP for Clinical and Translational Science, all at Stony Brook University School of Medicine. She did her medical training actually in Lebanon at St. Joseph's University, and then came to the United States, where she did her residency in Memphis, Tennessee, then did a fellowship at NIMH, and then had a faculty position at Yale, was then at Columbia University Medical Center for many years, and in 2016 went to Stony Brook University. She is an internationally recognized expert in a few different areas, including molecular imaging and PET imaging, pharmacology, schizophrenia, and addiction. Her work centers on looking at molecular markers and their significance as measured with multimodal imaging approaches in psychiatric disorders. She's been funded throughout her career extensively through NIMH, NIDA, NIAAA, and various other industry and foundation sources. Her works really advanced our understanding of, among other things, the nature of dysregulated dopamine in schizophrenia. And I would say she's really done a lot to revise the field's understanding of how dopamine is dysregulated in schizophrenia, beyond the typical dopamine hypothesis that everybody learns about. So we'll be hearing about that today. Okay, so that's, I want to leave some time for her. So please welcome Dr. Anissa Abi-Dargham.
Good morning, everyone. Good afternoon. Can you hear me? Okay. I want to thank Steve for the kind introduction and also for the invitation. It's really special to give the Henry Nasrallah lecture, considering that we come from the same country, so it's particularly special for me to be here. I've had friends at University of Rochester and also previous mentees and colleagues here, so this is really really pleasant to be with you today. So as Steve said, I'm gonna give you, share with you a little bit about our studies spanning a few decades now, looking at the neurocircuitry and the neurobiology of the brain in schizophrenia. We concentrated initially on dopamine, but then has taken us into different directions. And I'll tell you why we concentrated initially on dopamine and how we attempted to study it and take you through the results we've had. These are my disclosures. Okay. Can you see the screen? Okay. So for those of you who study schizophrenia, you're probably very aware of how many functional domains are affected in this illness. the behavioral manifestations are really very widespread. You have perceptual abnormalities with psychosis, hallucinations, paranoia, thought disorders. You have cognitive deficits, and these kind of encompass multiple cognitive domains, things that are very different from each other, like social cognition versus executive function, subserved by different parts of the brain. Movement abnormalities that were described way before antipsychotics were used. Those are known to induce motor abnormalities, but we know in schizophrenia there's like catatonia and stereotypies that were described by Kraepelin way before antipsychotics were used. Mood disturbances, reward deficits such as anhedonia. This really suggests that almost all systems in the brain are affected. They say global brain disorder. So what could explain something like this? It could be that multiple brain regions are affected, or it could be that a linking mechanism, a circuit that kind of connects these regions together is affected, has a central lesion, and that is reverberating across all these functional domains. So these are the kind of questions that we're grappling with over the years that we tried to address. Just for those of you who don't look at brain scans often, I just want to introduce some of the regions that I'm going to be focusing on. You see, this is a coronal section of the brain and a sagittal section near the lateral ventricles. You see the caudate, putamen, and between the ventral striatum. These have different functions. The ventral striatum houses the nucleus accumbens that deals with emotions. You see the temporal cortex down here. Are you seeing my cursor? Okay, good. The frontal cortex, oops. And you see the thalamus. And I wanted to point out here in the midbrain, the area substantia nigra and ventral tegmental area that houses dopamine cells. This is where dopamine cells originate, and from there they project throughout the brain. Now, these are reorganized in this cartoonish way, and I'm going to be referring back to this cartoon throughout my talk to make it easier to remember the studies and the findings. And these brain regions are linked by circuitry that we call the cortico-striato-palido-thalamocortical loop. So you have projections from the cortex that are glutamatergic in green down to the striatum. These are glutamatergic projections. From the striatum, you have GABAergic projections going from the striatum to the globus pallidum, from the pallidum to the thalamus, and the thalamus projects back to the cortex via glutamatergic pyramidal projections. All these loops are heavily modulated by dopamine. In blue, you see the dopaminergic projections with the Every time I move this, it advances the slide, so I'm trying to be careful. So I'm going to just not use the cursor. So you have the projections to the cortex, that the mesocortical projection. In the cortex, D1, mostly one of the dopaminergic receptors that is heavily involved in basically the impact of dopamine on the circuitry, but you also have D2 and D4. Then you have the mesolimbic projection, going to the ventral striatum. Actually, in the striatum, you have as many D1 as D2, but you also have some D3 receptors. And you have the nigrostriatal projection that goes to the dorsal part of the striatum. These, the dopaminergic projections modulate these basal, cortico-basal ganglia loops in very important ways. For example, in the cortex, dopamine is known to work via D1 and D2 receptors to some extent to affect the inhibition to excitation balance in the circuitry in the prefrontal cortex, which is critical for cognition. So a major effect of dopamine via D1 and D2 on cognition, the prefrontal cortex. In the midbrain, the VTA, it's been shown that dopamine is involved in reward processing. So for example, you see here, this is from recordings in rodents when they're being given unexpectedly a reward, like a sugar pellet. You see firing of dopamine cells. This is this heavy band. Then the firing of dopamine cells moves to the cue that predicts the reward. And if the cue does not result in a reward, then you have silencing of dopamine cells. So dopamine cells are tracking the mismatch between the expectations and the experience. We call this prediction error. It's either a positive prediction error, the experience is better than the expectation, or negative, the experience is worse than the expectation. In addition, dopamine modulates the outflow from the striatum onto the rest of the circuitry via the GABAergic projections that are organized in D1 and D2 pathways. So the orange circuitry here is the direct pathway. We call it also the GO. Because of the way the circuitry works, you have the GABAergic projection and pallidum onto a GABAergic projection that goes to the thalamus. So you have inhibition on the inhibition, which basically releases the thalamocortical communication, facilitates it. While with the red D2 indirect no-go pathway, the circuitry works so that you have more of an inhibitory effect on this thalamocortical projection. Actually, can I have that pointer? Because I think I'm going to point on the slide, because if I touch here, it moves it. And this will do it? Yeah. Okay. So you see here, you have the inhibition onto an inhibition which releases the stimulation onto the inhibition back. So basically now you have an inhibition from the no-go pathway and a stimulation from the go pathway. And so the thalamus is always under this kind of balance of go, no-go, approach or avoidance that dictates our behavior. In addition, dopamine modulates this go, no-go. So dopamine will activate the go and inhibit the no-go. So if you have excess dopamine in the striatum, you can think about it as there's no break. That's all acceleration. Now, another thing I have to tell you about is that the striatum is very diverse in its function. So when you go from anterior to posterior, you're moving from things like the ventral striatum, which are all limbic, emotional processing areas, to the associative striatum in yellow, anterior caudate, anterior putamen, posterior caudate, that are cognitive. And that's because of the kind of inputs they receive and what they project. And then you have the sensory motor striatum, which is the posterior part of the putamen, in blue, that have to do more with sensory motor information. These different parts of the striatum are connected to different parts of the cortex and project to different parts of the rest of the circuitry that I showed you, so that you kind of have segregated, topographically segregated loops among these corticobasal ganglia loops that have different functions. The limbic loops, associative loops, sensory motor loops. So basically, dopamine is involved in modulating these loops that have a variety of functions across multiple domains. And these are the kind of functions that are abnormal in schizophrenia. So it's kind of very relevant to the different type of symptomatology that we see in patients. In addition, it was well known at the time when I started my career that all drugs that we use to treat schizophrenia are D2 drugs. And not only they're D2 drugs, their affinity at the D2 dictates their clinical dose, which means you really need to get to that D2 receptor enough to be able to have a therapeutic effect, which really implicates the D2 receptor in the treatment of psychosis. There's one exception at this point to this rule, which is this new muscarinic drug, xanomeline trospium, Cobenfy, that you probably all know about. So after having established that dopamine is relevant to the symptoms and to the treatment and is critical to the circuitry that observes functions that are altered in schizophrenia, then the next question is, so how do we study it? Well, we use a multimodal imaging approach. We actually even collect DNA to, I will not be talking about this, to calculate the PRS, the polygenic risk scores for each patient. We get molecular information with PET, positron emission tomography. I'll tell you a bit about it. Magnetic resonance spectroscopy, neuromelanin sensitive MRI. This can help us to try to address mechanisms and consequences of things we observe in patients by going into different systems, such as, you see something in patients, you could go into an animal model or even other preclinical models to try to understand something like what we observe could be a result of what other abnormalities and lead to what abnormalities. Then we obtain functional information on every patient that goes through the molecular imaging, that is with fMRI, task, and resting state to be able to link the molecular and the functional. We do very extensive clinical evaluations, cognitive evaluations, and even evaluation of sensory processing, so we can have a full picture on every patient that goes through the studies. And the point of all that is to get information on targets, therapeutic targets, and potential biomarkers. And I'll touch on those at the end of the talk. So I will touch on the pharmacological targets, especially the more recent work, not just dopamine, but also the cholinergic system and kappa opioid receptor system. But also how the circuitry abnormalities that are linked to these molecular abnormalities can be targeted with neuromodulation techniques, and also highlight some of the cognitive basic computational abnormalities that could one day maybe be addressed with computational approaches. So this is a cartoonish representation of the PET molecular imaging technique. We have, that depends on really basically coming up with a radio tracer. That is a ligand that is specific and selective for a target in the brain that you'd be interested in. It's made radioactive with a radiochemist putting a radioisotope onto the ligand. So now it's a radio tracer. It's injected into a subject laying down in the scanner. It will flow throughout the body and the brain, but then concentrate on the sites in the brain that it is specific and selective for. The scanner will obtain the radioactive signal, and that is basically what leads to the radioactive image here. We obtain an MRI on every person to be able to more precisely draw the anatomy of the regions we're interested in. Then we have two important pieces of information. The input function, which is calculating, measuring in the blood how much of the radio tracer we injected is present, is not metabolized, is not bound to protein, because that's really what crosses the blood-brain barrier. That is what the brain is exposed to. And then we measure what the brain response is with this radioactivity over time in the brain regions, as time activity curves, putting together the input function with the response of the brain. Through mathematical modeling programs, we can obtain the outcome measure of interest, which is what we call binding potential, or something similar to that, which basically tells you how many receptors are in a brain region. So using these techniques, we developed or used tracers to look at the dopaminergic synapse. I want to start with the striatum. And for the striatum, we have like kind of lots of richness in a way, because we have lots of tracers for different aspects of the synaptic machinery. So we have, for example, a F-DOPA, which is a radioactive form of DOPA, which when you inject basically goes through the cellular machinery as if it was DOPA. So you kind of have generation of F18 labeled dopamine, and that's what gives you the signal that the PET scanner will detect. So it's a rate of dopamine synthesis, it's the activity of this AADC enzyme, transforming DOPA into dopamine. We also have radio tracers for the vesicular monoamine transporter, which is this transporter that's on the vesicle that brings in dopamine. Inside the vesicle, dopamine is those red circles, pink circles, because you want to preserve dopamine in a vesicle for when you need it, when there is an action potential. If dopamine is in the cytoplasm of this presynaptic dopaminergic cell, then it will be metabolized by COMT and monoamine oxidase. We have tracers for the dopamine transporter. That's the Hoover of the system. This is what cleans up dopamine from the synapse, takes it back inside the presynaptic nerve terminal. We have tracers for dopaminergic receptors, such as D2 and also D1 and the dopamine transporter. We devised, in addition to that, ways of measuring dopamine itself. And the way we measure dopamine itself is through kind of a subtraction technique. We obtain a first D2 scan by injecting a D2 antagonist, a D2 radio tracer. The radio tracer is in blue, is going to bind to the D2 receptors that are here in pink. Dopamine is in red, some of it is flowing inside the synapse between the two nerve cells. And so then we disturb this equilibrium by doing two different types of paradigms. One of them is injecting a psychostimulant that will break down the vesicles and dump dopamine in the synapse. Now you have competition between dopamine and the radio tracer for binding to the D2 receptor. So the binding potential or the radioactive signal is going to go down in the second scan because you have a lot more of the red dopamine binding over the blue radio tracer. So that is kind of a measure of how much dopamine was stored in this presynaptic nerve terminal. And we have the opposite manipulation, which is deplete dopamine completely. Give a blocker of tyrosine hydroxylase, the rate-limiting step in dopamine synthesis, over 2 days, so that you get rid of all the dopamine. Everything that's in the synapse now is gone, all the red is gone. And comparing this to the first baseline scan tells you how much dopamine was in the synapse to start with. So this is like a measure of D2 occupancy by dopamine. We applied all these to patients with schizophrenia and controls, patients who are drug-free. Obviously, you have to have the D2 receptor be available for the radio tracer. And so now I'm going to tell you what one additional point, technical point, which is you could use different D2 radio tracers that are optimized for different parts of the brain. So for example, raclopride shows you best the striatum, D2 and the striatum. If you want information on D2 and dopamine outside of the striatum, that's not going to help you. The signal is too low. Then you use something like FLB 457, and I'll show you results with that. It will allow you to look at cortical and extrastriatal regions. And the striatum, although it lights up, it's not usable because you don't get to equilibrium during the scan. And without equilibrium, you cannot quantify the signal. Equilibrium means, this point where you have association between the tracer and the receptor reached like a peak and then it starts to come down. If you don't capture that curve, you cannot analyze the data. So with the striatum, because there is so much D2 receptor and the tracer has higher affinity, it keeps going up and up. We also used a PHNO tracer recently because it allows us to look at both striatum and some extrastriatal sites, like the midbrain, substantia nigra, see it here, globus pallidum, thalamus, So there was a way of looking at both in the same patients, in the same session. The additional thing I wanted to tell you is that tracers could be antagonists or agonists. Antagonists bind to the whole pool of D2 receptors, the light blue and dark blue, D2 high affinity, D2 low affinity state, or they could be the agonist binds only to the high affinity state. And that has impacts also on the kind of results that we can obtain. Okay, so what did we see in the striatum using all these technologies that I've just described to you? This is a summary slide from our own studies, but studies from the field in general, showing that dopamine synthesis is increased, dopamine release is increased, storage capacity, dopamine occupancy, D2 occupancy by dopamine, synaptic dopamine is increased. D2 receptors are increased only in patients who have received antipsychotics before, not in a drug-naive state. D1 receptors in the striatum, the dopamine transporter, and the vesicular monoamine transporter are normal. When we pool these studies together, we get an effect size of about 0.8, which for psychiatry is a big effect size, but unfortunately might not be enough to lead to clinical utility, as we were discussing last night at dinner. So there is definitely an increase in presynaptic dopamine and schizophrenia in the striatum. Now, what does that mean in terms of symptoms? Well, we've established that it's related to psychosis. It's related to the response of psychosis to our D2 drugs, which is normal. If dopamine is increased in a striatum causing psychosis and you give someone a D2 blocker, it's going to improve it. This was replicated in a different paradigm, a different lab, by showing that F-DOPA is increased in treatment responders, but not in treatment resistance. Those have similar levels to controls. So basically, if you have schizophrenia, psychosis, and increased dopamine in the striatum at the same time, you're lucky because we have drugs for you. Now we looked within the striatum, since the striatum is so varied and has so many different functional jobs to do, where is the biggest dysregulation? And we were surprised to find that it's actually in the associative striatum, not in the ventral striatum, as people think about the mesolimbic hypothesis of schizophrenia. Well, this really suggests is the nigrostriatal hypothesis of schizophrenia. Not to say that dopamine is not increased throughout. Here, the patients are in red, the controls are in black. This is the amount of dopamine that was kind of uncovered in the synapse. And you see that the biggest effect is in the head of the caudate. This is like the cognitive striatum, the middle section of the striatum. In the ventral striatum, although dopamine levels may be elevated, low dopamine related to severity of negative symptoms. Obviously, this is only one small observation needs to be replicated, but just to kind of tell you that, you know, in the ventral striatum, if you have maybe excess dopamine, that contributes to psychosis, but a deficit in dopamine will contribute to anhedonia, amotivation, etc. So just to summarize what I just said and putting it back onto the functional subdivisions. Now, it may not all be presynaptic. We have here this beautiful work by Dr. Judy Thompson that was very influential, actually, in modifying how I think about dopamine in the striatum and schizophrenia. Most people will say it's a presynaptic increase. Based on this study, I think it could be also something abnormal about the D2 receptor itself. And the reason we say that is that this was a group that had dual diagnosis, schizophrenia and addiction. The addiction dampens dopamine. That's known, you know, Nora Volkow's work, other people, alcoholism, cocaine, opiate use. In addiction, you have low dopamine if you use these paradigms. So this group had low dopamine because the addiction kind of like trumped the schizophrenia in a way. And they're all below normal levels of dopamine release. But look at the relationship with psychosis. They still had the minute changes in dopamine stimulation of D2 led to an increase in psychosis, kind of signifying that there are factors related to D2 itself. We call this postsynaptic factors. We're still studying those. Now, I'm going to tell you very quickly how we address the question of what's the impact, the functional impact of excess dopamine in the striatum on these functions that are affected, such as, perceptual abnormalities and others. How do we go from having excess dopamine in the striatum to hearing voices, for example? And I was fortunate to have someone at that time at Columbia in my lab, Guillermo Harga, who's an expert on bias perception. So we learned a lot about that, but we also were able to combine these studies from very different kind of points of view. So I have to tell you a little bit about perception. Perception is not what we think is just, and many of you are very much like experts on this, so my apologies, but for most people, maybe we think that a stimulus, whether it's a noise or maybe something else, is just kind of registered as it is. And the reality is really an integration between what we expect to hear and what the sound is. And that has to do, for example, with, how precise the sound is, how clear it is, or the context, what we're expecting to hear. So there is always this kind of prediction that, then there is the experience, and that leads to computing a prediction error. So how close was the experience to what we expected to experience? The prediction error on a trial-by-trial basis is used unconsciously, obviously, by our neurons in the brain to update the next prediction. We call this scaling or weighing of the prediction of the mismatch, the prediction error. And we can measure this by following the fMRI signal, how closely it matches this prediction error updating. How much is it paying attention and, changing? And so our question was, by combining those kind of like fMRI studies of bias perception with dopamine imaging in the same subjects, we could address the question, how is dopamine related to those phenomena of bias perception? So the first study we did was an auditory task where people are exposed to different duration of auditory tone. And I will tell you more about it in the next slide. But just to illustrate what I just showed you here, what we're looking at is, you are kind of sitting around having some kind of expectations. You could have very strong expectations of hearing something or weak ones. Then you have the stimulus itself. And then your experience is going to be very much affected by the integration of these two. If you have strong expectations in one direction or another, we call this strong priors, your experience will be very much affected by that. If you have no particular expectations, you're going to have a more accurate recording of the stimulus. And this is now making it more concrete. The task was basically exposing people to auditory stimuli and then exposing them to a target auditory stimulus and asking them to rate how long it is. If the context was very predictable with short tones, the target auditory tone was judged as shorter than it actually is. If the preceding tones were variable, sometimes long, sometimes short, then the target auditory tone was experienced more accurately for as long as it is. So we call this the uncertainty effect. If the context is uncertain, you don't have strong expectations. You don't have strong priors because, it's uncertain. And so this uncertainty effect was weaker in patients with schizophrenia. And now we could ask, so how does that relate to dopamine? And what was really interesting, and this is one of the most exciting studies I think we've done, is that we found first that the weaker the uncertainty effect, the more hallucinations, but also the weaker the uncertainty effect, the more dopamine and the striatum as a whole, and then the associative striatum. So dopamine is really interfering with the coding of uncertainty and at a very basic cognitive level of auditory processing. Just to summarize it, high dopamine in the striatum weakens the uncertainty adjustments, which means increases the expectation biases, which leads to biased perception and hallucinations. So that is like, a basic cognitive mechanism that could in the future potentially be addressed if we know how. We're delving deeper into this, and we've just finished a much bigger study to look at both sides of the equation, not just the effect of priors and uncertainty, but also updating the prediction errors and deficits in this updating, and looking at what are the mechanisms of this biased inference or biased perception. 3 major hypotheses in the field. One of them is the one we like, which is that abnormal dopamine in the striatum is affecting the outflow, the circuitry from the striatum onto the rest of the circuitry, and that's really biasing, producing this bias, just because of the finding I just described. Other people think it's an NMDA glutamatergic problem just within the cortex itself that makes the cortex maybe hyperstimulated, and that's why schizophrenia patients have activity in the auditory cortex during silence, is because it's just excess activity and it's being kind of encoded as if it was an outside stimulus and a hallucination is experienced. Other hypotheses have to do with the input from the thalamus. So we put in all this together in a major study, multidisciplinary, many collaborators, and I'll show you the results only from one piece of it today, but we put together auditory processing task with thalamo-cortical connectivity, but focus on the medial geniculate nucleus where auditory processing happens, so more precise. And looking at glutamate and the cortex, the auditory cortex so these are, Guillermo Harga at Columbia, Christophe Ucam, now in Zurich. And then the PET, looking at dopamine release and striatal and extrastriatal regions. Initial results indicate that dopamine in the striatum is the biggest culprit, but I will not show this here today. So now, what about the other functional effect of excess dopamine in the striatum? And one major readout is connectivity, of course. And I'll show you only one study that was done very early on in these patients, looking at the connectivity of the striatal subdivisions to the rest of the cortex. And you see here in blue is the head of the caudate. That is the region that has the highest dopamine dysregulation. And you see that in patients, the head of the caudate has the weakest, has less connectivity to the rest of the cortex and the brain than in controls. In controls, the head of the caudate is the most connected subdivision of the striatum. It's almost like the maestro of operations. But that kind of like prominent role is lost in patients. And that relates to dopamine in that region and to psychosis. So just to summarize until now, excess dopamine in the striatum is related to perceptual bias, psychosis, response of psychosis to antipsychotics, and altered connectivity. Now, I showed you a lot of data, but this is really the most important kind of conclusion. It's really messing up with the functions across domains that you may not have expected in a way. So now, what about cortical dopamine? For that, we had to go to another tracer, as I mentioned. So we do a PET scan before and after amphetamine to look at dopamine release and extrastriatal regions. And we do fMRI during working memory to be able to look at cortical dopamine, obviously our most direct relevance here is the effects on cognition. And so the study showed, and that's been published a few years ago now. So you see in the dorsolateral prefrontal cortex, patients are here in red and blue, compared to controls in black. Patients show less dopamine release. So unlike what I've just showed you in the striatum so extensively, in the cortex is the opposite. They have less dopamine release using these same paradigms. And this dopamine release in both groups is related to how well they do during the fMRI task on working memory. So the more dopamine they have, the more they can activate their prefrontal cortex and perform better in a way. In addition, in a small group that also did the NBAC task, in patients with schizophrenia, we found this incredible relationship between dopamine release itself and NBAC performance, which is really difficult to capture. And, this is not necessarily it means that you can find this relationship no matter what you do, no matter what working memory task you use, because we didn't see it with the other working memory task. But just to be able to see it even with one just indicates that really dopamine may be involved very intimately with working memory via its activity in the prefrontal cortex. So this was the one result we were looking for. It's what's going on in the cortex in patients with schizophrenia with dopamine. But what was really stunning is what we also found in other regions. In black here is the displacement seen in controls, and in red in patients. I've shown you the DLPFC, dorsolateral prefrontal cortex, which is involved in cognition. But look at the other cortical regions, same pattern. Amygdala, hippocampus, again, same pattern. Even the substantia nigra VTA, midbrain dopamine cells, same pattern. So patients show less dopamine release than controls. So this kind of implies that the brain in patients with schizophrenia in general is hypodopaminergic with this kind of more localized increase that is just in the striatum, which is kind of really puzzling as to how could this happen. And this really took us into the next phase of studies we've done to try to look at this almost opposite dysregulation, because the dopaminergic projections are coming from the midbrain. So how is it that you could have less release here, the release you measure at the somatodendritic level, but yet more release in the striatum? Now, it could be that there are just factors related to the dopaminergic neuron itself, where there's something in the projection and the striatum that's dysregulated. Or it could be that our modulators over dopamine and the striatum that are affecting terminal release. So we're looking at both aspects. Now, the third question was, what about striatal and extrastriatal regions in the same patients? Can we measure this to see how are they related? And so we use PHNO for that. We just kind of finished the study. We're about to publish it, 29 patients, 28 controls. And here you can look at the striatum as well as the midbrain in the same scan. Do this before and after amphetamine again. And what we found is the following. This is the correlation between dopamine release in the midbrain and the substantia nigra and dopamine release in the striatum. And you see how much weaker it is in patients compared to controls. So basically what it means is that the midbrain, where dopamine cells activity is happening, is not really in sync with the terminal release in the striatum. in patients the way it should be. So there are other things happening that are kind of messing up this nice relationship where you have activity, it should correspond to what happens in the terminal field and projection. So what could be happening? We don't have answers, but we have initial questions that we've, trying to address, and I'll just show you this part of it. We got interested in the cholinergic system and then the kappa opioid receptor system. And because we're short on time, I'm just gonna go through this with a bit of just cutting down to the results. What we found at this point, looking at the vesicular cholinergic transporter, is that in patients with schizophrenia and the prefrontal cortex, more pathology, as you see here, was more psychosis or more cognitive deficits, relates positively to the vesicular cholinergic transporter. So the more vesicles, the more transporter, possibly meaning the more acetylcholine, was, patients are sicker. And that was not the case in controls. This is obviously a first observation. Jody just has now an R01 to replicate this in a bigger cohort. And we can discuss what the implications of that. For the kappa opioid receptor, this is one of the opioid receptors that is mu, that you're all familiar with, was like opioid agonists that make people have respiratory depression, like fentanyl, etc. The delta and the kappa. Kappa agonists make people psychotic, and kappa antagonists can actually help with psychosis. We did a meta-analysis that showed that all opioid pan antagonists, including buprenorphine, which has a mu agonist effect, but a kappa antagonist effect. So what they share is the kappa antagonism, can have an antipsychotic effect. So we thought, well, let's look at the kappa receptor. Maybe it's increased in schizophrenia, that's driving dopamine, and that excess stimulation is making people psychotic. And what we found is not consistent with our hypothesis. There was no group difference in numbers. There was no relationship to psychosis, but there was a relationship to anhedonia. The more kappa receptors, the more anhedonia in those patients. And this is kind of interesting because, many pharmaceutical companies are pursuing kappa antagonists for anhedonia and depression. I'm not sure that they're all successful at this point, but we think there could be potential for kappa antagonism for negative symptoms in schizophrenia. And we're pursuing this with an R01 submission that we hope will get reviewed at least. Okay, so, and this is the final kind of piece, and I'll wrap up in 2 minutes, just to take you into targets and biomarkers. One biomarker development that we are interested in that resulted from our common interest in dopamine and expertise in MRI is to look at the metabolism of dopamine, and that accumulates in the substantia nigra and can be detected with MRI. So basically dopamine, when it's dumped in the cytoplasm, is oxidized, binds to ferritin, and then has, gets polymerized and packed into organelles that will travel back to the midbrain and sit in those, substantia nigra and VTA cells. And there can give a signal that can be detected with an MRI. So we call this neuromelanin sensitive MRI. It's a specific kind of sequence. And what we've shown is that the signal detected with an MRI in dopamine cells and the signal that's measured in the striatum with PET are related by doing studies in patients who did the same, both modalities. So this is the neuromelanin signal versus the gold standard of dopamine release. They're correlated. There's been lots of publications. I'm not going in details, but just to kind of put it on your radar that this is potential biomarker that can index accumulation of dopamine metabolism over time. We have a multi-site study looking at D1 agonist in patients with schizophrenia for cognition that we just finished. It's multi-site at Columbia, Yale, Stony Brook, and UPenn, looking at acute administration, 5 different doses, and the readout is on the microcircuitry of the prefrontal cortex during a spatial working memory task. So basically, can we translate from this low dopamine into something therapeutic? Again, I won't show you results today because they're still under analysis. This Alan Anticevic was very instrumental in designing the study. And the final therapeutic comment I want to make is that we have one case only, but deep brain stimulation was a patient that had very resistant auditory hallucinations that nothing could address. And the deep brain stimulation was targeted to the substantia nigra, and this patient was cured. Like basically no voices. Actually so cured that he was almost missing the voices. He was like, it's too silent. And has been voices free since. So what does that mean? It means that abnormalities and, chemistry could affect the circuitry, and then possibly this could be reversed by, interventions that address this kind of like circuitry abnormality at any point of entry. Something to follow up on and test. Can we reverse these abnormalities by giving deep brain stimulation in the striatum or in the associative striatum? This was in the nigra, which wouldn't have been my first choice. How does that work? It's possible that either pharmacological interventions that block the D2 receptors or neuromodulation could reestablish this balance between the go, no go, and reestablish, more normal kind of flow across the circuitry. So with that, just want to leave you with the final graph that shows you all the results that I have shown you, linking the different functional domain to the different type of molecular abnormalities that we have measured. So the, psychosis could be related to D2 overstimulation and the associative striatum and possibly vesicular cholinergic transporter in the cortex. Cognition related to low D1 stimulation in the cortex and maybe abnormalities in cholinergic transmission in the cortex. The balance of D1, D2 outflow from the striatum could be critical and could be readdressed with neuromodulation. And finally, dopamine in the ventral striatum, as well as the kappa opioid receptor and limbic regions may be critical to anhedonia. With that, I want to thank many, many collaborators in my own group, but also across other institutions that have been very helpful. I've mentioned them as I went through the talk, and my funding agencies. And hopefully they continue to fund us, but also patients and their families for undergoing these very burdensome studies. Thank you. Five minutes.
All right, thank you. That was a fascinating talk. So much good information and updates. So let's see. Do we have any questions from the audience?
I hope that was clear. It was just, sorry.
Apparently it's so clear that there are no questions. But we do have a question that's come in over Zoom.
He's raising his hand.
Oh, Brian?
Thanks for the talk. There's neuroimaging biomarkers of the striatum. You and I were talking a little bit about this, where the biomarker is much more apparent for non-affective psychosis than affective psychosis. I was wondering if you looked at that distinction in your own data, looking at like dopamine synthesis and release capacity.
Yeah, so we're not looking in the sense that you're asking at affective versus non-affective psychosis, although I think this is very promising. But we are interested in connectivity per se, and what is the exact underlying piece that's critical. Because when you measure connectivity between A and B, it may be that there are sections in between that are abnormal, and the readout is kind of not looking at the precise intermediate nodes, right? So we initially looked at, we collaborated with Eric Kandel's group, where they produce a mouse that has excess D2 stimulation and a striatum by increasing the number of D2 receptors. And then they found that there was changes in the hardwiring of the circuitry and the projections from the pallidum to, outside of the pallidum. So between caudate to globus pallidum. Changes where the go is shifted to the no-go or vice versa, something like that. So we're looking at that in patients. I think there are multiple nodes of abnormalities and the striato circuitry connectivity, whether it's frontal cortex or other cortical regions. And it could be also within the basal ganglia itself. And so that's a very important area of work. And hopefully somebody will come to, be able to translate this into a usable way in the sense of a biomarker. But, as a lot of work is needed, multiple questions to be addressed. I think it's the same paradigm has to be used in a very systematic way, and it's big sample size to address, the biomarker type of questions. Ours are more mechanistic explorations.
Okay, so we only have about a minute, but in addition to this, all this work in dopamine, one of the other influential hypotheses of schizophrenia based on PCP and ketamine and all that is an NMDA receptor hypofunction. And how does what you talked about on these structures like the striatum, do you think, relate to those findings?
Yeah. So I think that, nobody has done really a very good work to address the relationships. I think there have been few studies that have linked glutamate by MRS in the hippocampus to maybe F-dopa in the striatum. The samples were small. I'm not very convinced that the data exists out there. Not that it's not happening, it's just that hasn't been done in a way that's convincing yet or persuasive. I mean, the glutamate system has got to be involved. I think the issue is we don't have good tracers to image what we need to be imaging. The MRS way of measuring glutamate and maybe GABA, I'm not sure is really the best way to address that. So for example, we don't see a difference in glutamate levels in the study I mentioned in the auditory cortex using MRS. Does that mean it's not there? It just means that our tool is too blunt. It's not precise enough. I have tissue glutamate. We're not looking at synaptic glutamate. We're not looking at the NMDA receptor itself. We don't have PET tracers. So I think that these questions have not been addressed convincingly yet using imaging and living patients. I would suspect that there's got to be involved. I mean, when I said the cholinergic system and the kappa opioid receptor system, we started to look at how could they affect dopamine. If I had the tool, I would look at the glutamate's effect on dopamine. So I don't have a good definitive answer. My gut feeling is it's got to be involved, but we don't have the right tool. Do we have the right tools, you think? I mean, is there something out there that we're missing?
I don't know. That's why I asked you. I figured if anybody knew, it would be you. But yeah, I haven't seen any convincing data on that. But yeah, that's interesting.
Yeah, more to be.
Okay, well, there's still a lot to be done. So I want to thank you all for coming. Remember, please to fill out the evaluation.
Oh, there's a question up there.
Yes, but unfortunately, we're over time. Denise, maybe you could come up and... all right. Okay, I'll allow it this one time. Very quick, yes or no question.
So thank you so much for presentation. My question was related to part of the blood-brain barrier. So nowadays we were interested with some mechanism of the drugs with my PI, Dr. Lizano. So if blood-brain barrier dysfunction changes how immune factors enter the brain. So I was wondering if it can change or shift D2 receptors sensitivity, or it can help explain why small dopamine change triggers the symptoms of the schizophrenia.
So how it changes across the blood-brain barrier could impact receptors and transmitters?
Yeah, if the blood-brain barrier dysfunction changes how immune factor enter the brain. So I was wondering it can shift or change the D2 receptor sensitivity. So it can be related to the symptom of the schizophrenia.
Yeah, do you want to repeat the question for me to make sure?
I think she's saying, can the disturbance in the blood-brain barrier, which is known to exist in schizophrenia and lead to inflammation and other things, can that affect the D2 receptors and maybe have caused some of the things you talked about?
Yeah. I mean, I would certainly say yes, because we know that toxic influences, drugs, famine, all kinds of environmental factors that happen like during development basically shape how the brain develops, and it affects the brain in multiple ways. So, for example, the D2 overexpressing mouse had, the increase in D2 was like during development and then turned off. But then the circuitry developed very abnormally in multiple parts of the brain. There's interaction between the inflammatory factors and, BDNF and things like that on the integrity and the growth of the circuitry. I would imagine these are very related. And I think there have been studies linking or trying to attempt to link dopamine development to inflammation. Maybe there's a study by Oliver House's group. But whether the data exists or not, I would think it's a very important question to address.
Okay. We really have to stop now. So thank you all for coming and have a great day.